Hello dear students, our today's topic is a very interesting topic and it is called as anti-ulcer drugs. More commonly they are also referred as antacids, and these are the H2 antagonist and the proton pump inhibitors which are under the category of anti-ulcer drugs. So let's move on to the first category of drugs, that are the H2 antagonist. As I mentioned in my previous lectures, the H2 antagonists are that chemical compounds which antagonize the action of histamine on H2 receptors. These compounds block the acid secreting units which are nothing but the parietal cells which are present in the oxyntic glands in the stomach. Here you can view the image. This is the oxyntic gland, and these green cells, these are the parietal cells which are involved in the acid secretion. So whenever there is increased acid secretion, there might be the severe conditions like peptic ulcer, the Zollinger-Ellison syndrome, GERD, that is gastroesophageal reflux disease. And due to this, there is urgent need of drugs which can revert the action of acid production. So it is found that the H2 antagonists are useful for treatment of such ulcerative conditions like peptic ulcer, Zollinger-Ellison syndrome, gastroesophageal reflux disease, which is shown in this picture. For some other conditions like stress ulcers and erosion. Stress ulcer is the acid production where the person is under stressful conditions. The erosion here stands for the production of some patches on the duodenal linings. So whatever condition they are very harmful for a particular person. So H2 antagonist, and for some other severe condition, the proton pump inhibitors are highly useful. Here you can view in this image that a person suffering from increased acid production may develop the condition where there is the red patches on the inside of stomach and the duodenum. Whenever this condition is achieved, there might be some reflux related to this, and this might result in vomiting, heartburn and other conditions. These are some causes of the duodenal ulcer. The first cause is infection by a gram-negative bacteria which is called as Helicobacter pylori. The other cause is the Zollinger-Ellison syndrome. The third cause is somewhat very common in our society, that is the high alcohol consumption. The next cause is also a very common one, that is chronic smoking. Now in today's life, due to some condition, there is increased stress and anxiety, due to which also there is increased production of acid. and this one is a drug induced acid production which is called as prolonged consumption of non-steroidal anti-inflammatory drug. Most of the anti-inflammatory drugs contains carboxylic acid. That's why after their consumption, the acid secretion might be increased. So that acid secretion will cause the ulcers, and to treat that conditions, the H2 antagonist and the proton pump inhibitors are highly useful. So let's move on to the first individual drug belonging to the category of H2 antagonist. The first drug is Cmetidine. Now dear student, if we look at the structure and the name, we can relate it with this structure. Here this C stands for the cyno, the im stands for imino, that is C double bond N. This Me stands for presence of the methyl group, and this 8 here stands for presence of this ethyl group, while this idine is used for this functional moiety which is called as guanidine. So due to presence of such structural feature, this compound has given the name Cmetidine. Now due to presence of this double bond, this compound exists in its geometric isomeric forms, that are Z and E. Now the therapeutically effective Cmetidine is the one having the E configuration. That means the group of higher priority on this carbon and the group of higher priority on this nitrogen are in opposite positions to each other. That's why this compound is called having the E configuration. Now this drug is short acting. That means its action will remain only for some time period. That's why its frequent dosing is required. And it is found that this drug alter the metabolism of other drugs, due to which this drug is not used for some other reasons, and that's why it is now replaced by the other potential drugs. Now one important point is that in Cmetidine the heterocyclic ring present is imidazole. Many times in GPAT examination there might be a question that the Cmetidine consists of following heterocycle. So 4 options will be given there and you have to answer that. It is imidazole, which is present in Cmetidine. Next, look at the next drug which is called as ranitidine. Dear students, once again you can correlate the name with this structure. Here this ran stands for this heterocycle furan. As I mentioned, in Cmetidine you have an imidazole ring, while in ranitidine there is presence of furan ring. Here this NIT nit stands for this nitro group, and this idine here stands for presence of this guanidine like portion, where the nitrogen of guanidine is now replaced by a carbon. The rest of the features are as shown in the structure. This drug is a very potent anti-ulcer and antacid drug. The bioavailability of this drug is around 50 to 60%. That means if 20 mg tablet is used, then about 10 to 12 mg of ranitidine will be biologically available. This drug is used for treatment of peptic ulcer. Now one important precaution which we have to keep in mind is that the antacid should not be used with ranitidine. That means ranitidine should not be combined or concurrently used with other antacids. Otherwise, the action of ranitidine will get decreased. A good example of use of ranitidine is its use with clarithromycin. The clarithromycin is a macrolide antibiotic, and this combination is used for treatment of duodenal ulcer caused by the gram-negative bacteria Helicobacter pylori. Next, move on to the next drug, which is called as famotidine. Just look at the difference. Here, the guanidine portion is retained and aminosulfonyl group is present on this nitrogen. The rest of the features are same here with a difference in heterocycle. In case of famotidine, the heterocyclic nucleus present is the thiazole ring, which is substituted with its position number 2. For thiazole, it is position number 1 and this is position number 2. So on position number 2, we have this diamino-methylene amino group at this position. So I repeat once again, dear students, that in case of cimetidine, the heterocyclic nucleus is imidazole. In case of ranitidine, it is furan, and in case of famotidine, it is thiazole ring. Like previous drug, this drug is also used for treatment of duodenal ulcer, the gastroesophageal reflux disease, and Zollinger-Ellison syndrome. Now, this drug is found to particularly inhibit the gastric secretions stimulated by food and the enzyme pentagastrin. Next, move on to the next category of anti-ulcer drugs, which are called as proton pump inhibitors, which are abbreviated as PPI. These drugs are used in treatment of duodenal ulcer, gastroesophageal reflux disease, Zollinger-Ellison syndrome, and some other ulcerative conditions. Now, this is a very important point. It is mechanism of action. These compounds, that means the proton pump inhibitors, are found to inhibit the pump which is called as hydrogen potassium ATPase, which is responsible for acid secretion from the parietal cells. Now, as it is, the proton pump is also made up of some proteins, and the ultimate units of protein are the individual amino acids. So, the proton pump inhibitors are found to bind with the cysteine amino acid, which is present on 812th site of the hydrogen potassium ATPase pump. So when the proton pump inhibitors bind with cysteine 812 amino acid of hydrogen potassium ATPase, there is inhibition of the acid production by this pump, and thus there is the decreased or ceased acid secretions. This is the general structure possessed by all the proton pump inhibitors. So that's why it is called as the general structure. So here we find that there is presence of two heterocyclic rings, like the first one is benzimidazole and the second one is pyridine ring. Both rings are attached by this sulfinyl methyl group. The sulfinyl group is attached on second position of benzimidazole, while the methyl group is attached on the second position of pyridine. That means both the rings are attached each other by their second site or second carbon by the sulfinyl methyl. So the common name given to this general structure is 2-pyridyl, because pyridine ring is attached with second position, 2-pyridyl methyl sulfonyl bench imidazole. The first drug is Omeprazole. If we know the basic ring structure, it is very easy to draw the structure for Omeprazole. Here this Ome stands for oxygen attached with a methyl. So here is the presence of methoxy group. So here is also a methoxy group, and this position we have a methoxy group which are surrounded by 2 methyl groups on this pyridine ring. Now dear student, if we look at the structure, we can find that there is no chiral carbon. But Omeprazole and the other proton pump inhibitors are available in their isomeric forms. Though we can wonder that why it is. It is due to the presence of this sulfonyl group. This sulfonyl group is present in such a way that this sulfur now has a tri-coordinated system. Tri-coordinated system means this sulfur is now attached with this benzimidazole, this methyl pyridyl ring, and the oxo group. So 3 main groups are attached. That's why the sulfur is having the tri-coordinated sulfonyl system. Beside this, it also has a pair of electron, due to which this compound, being consisting of the sulfonyl group, exists in the 2 isomeric forms, that is the S and the R enantiomers. Such kind of enantiomers we have undergone with the compounds having the chiral carbon. Now here in this case we have a chiral sulfur atom, due to which 2 isomers are possible, which are S and the R enantiomers. The therapeutically effective omeprazole is a racemate. That means it consists of both the S and the R isomers. As we mentioned in the previous slide, this compound is used for the duodenal ulcers and other ulcerative conditions. The next drug is Lansoprazole. Here also we have the same general structure, but one major difference is that one of the methoxy group is now substituted with ethoxy group, which consists of 3 fluorine on the second carbon. So this is the simple difference. Here is the absence of a methyl group, and instead we can say that one of the methyl group is migrated on this carbon and the hydrogens are replaced by the fluorines. This compound also is available in its racemic form due to presence of this tri-coordinated system by sulfonyl group. This compound is considered as the racemic mixture of dex-lansoprazole, that is the dextro isomer, and levo-lansoprazole, that is the levo isomer. The next drug is Rabeprazole. Now, if we compare this structure with the previous proton pump inhibitors, we find that again, instead of the methoxy group, we have a propoxy group over here. That means the oxygen attached with the 3 carbon chain, whose 3rd carbon is once again replaced by a methoxy group. So this is a very potent antacid drug. Now one very important point is that the omeprazole, lansoprazole, rabeprazole and pantoprazole, all the anti-ulcer drugs are available in 20 mg delayed release tablet form. Delayed release so that the drug can be released for longer period of time, and at the same time it can remain protected by the acidic environment in the stomach. The last drug is pantoprazole. Here the methoxy group is retained and one of the methyl group is replaced by another methoxy group. On benzimidazole ring structure at position number 6 we have a difluoromethoxy group. So ultimately the methoxy portion is increased, and due to this there is a good increase in the overall activity. This drug is also available in its racemate form. That means both the isomers, that is R and S isomers, are present in the compound. This drug is also available in 20 mg delayed release tablet form. So dear students, that is all about the anti-ulcer drugs containing the H2 antagonist and the proton pump inhibitors. Thanks for watching the video. Please like, share and subscribe. Thank you.